Hello again. When you hear virus, you may immediately think disease or infection. But what if I told you that viruses or virus-inspired tools may become powerful allies in restoring and protecting our environment? In this video, we explore how engineered viruses, virus-derived particles, or hybrid bio-nanotechnologies could be used for bioremediation, sustainable agriculture, and ecosystem repair. But of course, these approaches and applications are not without risks and ethical questions. So we'll examine both promise and caution. Viruses are marvels of precise nanoscales, engineering, protein shells, self-assembly, capacity to carry payloads. Researchers borrow these properties for the design of virus-like particles, as on the pictures you see, or modified viruses as delivery and functional agents. For instance, capsid proteins or viral scaffolds can be functionalized via chemicals methods or genetic engineering to carry enzymes, catalytic groups, or pollutant-binding moieties. In effect, you turn a virus architecture into a mini reactor or binder repurposed for environmental tasks. Let's dig into some concrete examples and promising directions currently explored in research. The first one concerns the phage biocontrol in agriculture. Bacteriophages, viruses that infect bacteria, are being explored as natural, targeted alternatives to chemical pesticides. A carefully chosen lytic phage can kill a plant pathogen without harming beneficial microbes, as you can see on this illustration. Some greenhouse or hydroponic systems test phage treatments in irrigation waters to suppress bacterial diseases with less chemical input at the end. The second example concerns a hybrid bio-nanotech virus scavengers. It's a fascinating recent innovation, engineering a single-celled organism, paramecium, by integrating magnetic nanoparticles decorated with virus-binding antibodies into its vacuoles, as you can see on this schematic illustration. This creates a semi-artificial organelle. We call it VSO, virus scavenging organelle, that captures waterborne viruses and chemically inactivates them inside the cell. Afterwards, the engineered paramecium can be magnetically retrieved, minimizing residual contamination. And because internal nanoparticles act as nanoperoxidase, it generates reactive radicals in acidic vacuolar conditions to destroy viral particles in situ. This is a bold hybrid of biology plus nanotechnology. As we saw with the previous example, the paramecium VSO system works as a virus-powered sustainability loop. Viruses help the planet, and other microorganisms help manage viruses. But it can be even better. When we look ahead, researchers imagine viruses or virus-derived systems that could degrade plastics in soil, detoxify heavy metals, or even restructure completely microbiological communities to restore ecosystem functions. These ideas remain in very early conceptual phases, but they inspire very much the new directions of research. As promising as all these ideas are, they come along with serious challenges and ethical concerns we must not ignore. Off-target effects and ecological disruption. A virus released in an environment might infect unintended hosts or spread beyond the intended area. Horizontal genes transfer. Engineered genes carried by vectors could be easily transferred to other microbes or organisms, creating unpredictable consequences. There are some other questions too. control and reversibility. How do you turn off or remove the viruses or modified agents once it has done its job? Biosafety and regulation. Stringent evaluation of these questions is required to ensure human, environmental, and microbiological safety before any real deployment. Scalability and cost. What work in the lab may be too costly or inefficient in real conditions? All these are questions to be considered. So finally, can viruses save the planet? The answer, possibly, but only if we proceed with care, creativity, and responsibility. Virus-inspired or virus-engineered tools could become powerful components in the toolbox of sustainable ecology, agriculture, and remediation, provided we rigorously address safety, regulation, and unintended consequences. For you, the next generation of scientists, reflect on this. What hidden function might viruses already play in nature, and how could we responsibly harness all those viruses for environmental good? Let's keep exploring the invisible threads that connect viruses, life, and our planet.